The overall goal of this procedure is to quantify cortical folding or gyrification with an exquisite resolution based on cerebral MRI. This is accomplished by first creating accurate three-dimensional representations of the cortical surfaces using the FreeSurfer software. The second step is to construct an outer surface tightly warping the pile surface using a morphological closing algorithm. Next, hundreds of overlapping circular regions are created over the outer surface and are matched to their corresponding patch of cortical area. Then the ratio of both corresponding regions is calculated at each point, resulting in individual maps of local gyrification, which can be subsequently used for statistical comparisons between different diagnostic groups. Ultimately, measurement of cortical structure in grown-up children, adolescents, or adults can uncover abnormal cortical development that occurred in the first months of life.
The main advantage of this method compared to existing techniques such as curvature-based or sulcal morphometry is that it provides an easily interpretable measurement of the gyrification, which is unrestrained by the sulcal walls and also offers exquisite resolution.
This method can help us answer some key questions for the development of psychiatric condition or neurodevelopmental disorder. In other words, we can understand how very early neurodevelopmental events, cortical folding, can later on impact the development of pathogenesis in adulthood.
We first had the idea of this technique when we realized that the gyrification index that was used in comparative neuroanatomy since decades could be a very powerful indicator of the brain development if we could overcome the issues in the way it was implemented.
The method will be demonstrated to you.